In this video, I want to tell you about the work we have been doing in the Lead Lab to design better vaccines against viruses like influenza, HIV, and SARS-CoV-2. In this video, we will focus specifically on influenza. First, we'll discuss an overview of your body's B-cell immune response to viral pathogens. Then, we'll talk about how influenza can evade your immune system, making vaccines less effective. Finally, we'll learn how we can hijack viral immune evasion to design more effective vaccines. Let's start by looking at a B-cell immune response. First, a virus enters the body. Then, an antigen on the surface of the virus binds to a receptor on a B-cell. This activates the B-cell, which then becomes an antibody-producing cell. The antibodies produced bind to the antigens. Once the antibodies are bound, the virus can be cleared by immune cells. Now that we know how influenza can be captured by the immune system, let's learn how it can evade your immune system. In pink is an antibody against influenza. That was this drawing in the cartoon. Here is an influenza antigen. In the cartoon, those were the spikes on the virus. Antibodies bind to specific regions on the antigen. Here, the pink antibody will bind to the pink region. To block the antibody from binding, the antigen can mutate. One way the antigen mutates is to add a sugar, known as a glycan, to its surface. Then, the antibody can't bind. The influenza antigen has many of these glycans on its surface. Over time, the antigen has mutated to cover different regions. These changes allow the virus to evade your immune system because antibodies that bind one form of the antigen will be blocked from binding to another. This is why you must get a new flu shot every flu season. Fortunately, some regions on the antigen aren't blocked or mutated. These regions are typically hidden from selective pressure to change, or they are important for the structure and function of the antigen. These regions are conserved across many forms of the antigen. While these regions are typically hidden, under certain conditions, the region is exposed. Once this region is exposed, an antibody can bind to it. Antibodies that bind this conserved region can bind to many forms of the antigen. We want to find these antibodies so that we can design vaccines that bind the antigen even when it mutates. That brings us to our third section on how we can hijack viral immune evasion to design more effective vaccines. Using biological engineering, we can add glycans to the surface of the antigen. This blocks all of the binding regions except the conserved one shown in blue. Now antibodies can only bind to this region of interest. To test if this engineered antigen can elicit antibodies that bind to the blue region, we will inject it into mice. As we saw earlier, the introduction of the antigen causes the mouse's B cells to produce antibodies that bind to different regions of the antigen. After we collect the antibodies, we can run the sample through our designed antigen and find which antibodies will bind to our region of interest. Once we know the designed antigen can elicit antibodies to the blue region, we can use it to design a more effective vaccine. Thank you for watching. If you enjoyed this video, you can visit the Lead Lab website.